In this webcast, we would like to outline the importance of sample preparation when using Merck Millipore's analytical test kits. We will also explain the most common sample preparation methods. These simple steps will facilitate your analytical work and ensure reliable results. Merck Millipore's test kits provide accurate results with minimal critical handling steps. This makes them a great tool for both advanced and occasional users. The test kits not only allow you to analyze all types of freshwater, wastewater, and seawater, but also beverages and solid material such as food and milk products. They can even be used to analyze soil after appropriate sample preparation. In all cases, correct sample preparation is key to minimizing errors in your analyses. A pre-check is an essential step which is performed prior to analysis in order to evaluate the sample type and ensure appropriate treatment. For example, non-homogeneous samples must be homogenized using a mixer or a homogenizer before further steps are carried out. Turbid samples must be filtered. Intensely colored samples should be decolored and then filtered. And pH levels must be checked and adjusted if necessary. Analyte concentration, or the concentration of interfering molecules, should also be determined, and the sample must be diluted to appropriate levels if required. If organics, oxidants, reductives, or complexing agents are present in the sample, further pretreatment and chemical digestion are necessary. Chemical digestion methods are described in a separate webcast in this series. Turbidity is caused by solid particles suspended in a solution. One of the simplest ways to clear such a solution is through filtration. Suitable samples can be filtered using simple paper filtration or centrifugation. For faster and more controlled handling, we recommend the use of a syringe filter with an adequate pore size. The choice of filter material is another important consideration, as it needs to be absolutely stable with respect to the chemicals used in the analysis, and must not dispense the analyte or add impurities that could interfere with the analysis. Filtration is not appropriate if your analyte is not water-soluble and is bound to the colloids that are causing the turbidity. Please read filter specifications carefully to be sure that they are suitable for your purpose. Intensely colored samples are common in the beverage industry, for example juices or red wine. These may often lead to false positive results in the photometric determination of the analyte, particularly if there is a co-absorption of the color at the wavelength of the photometric method being used. Hence, such samples must be decolored, for example by treatment with adsorbents such as activated carbon or polyvinyl polypyrrolidone, also known under the trade name Divergon. Divergon is used, for example, to prepare intensely red-colored juices before measuring ascorbic acid with the Reflectiquant system. For a more detailed look, please watch the video. Care should be taken to ensure that the adsorbent does not adsorb the analyte or set free interfering substances or the analyte. Dilution is necessary when the sample needs to be matched to the correct measuring range. Interfering substances can also be diluted to a concentration that no longer affects the analysis. In either case, it is important to use only high-grade distilled or deionized water. We recommend the use of ultra-pure water plants such as the Millipore Advantage System, M-Sure water for analysis, or the Lycrosolve water for chromatography. Water specifications must conform to test kit requirements. Always check the pack insert to ensure that your water is suitable. High chloride levels are present in many samples. This can interfere with measurements, for example when determining the chemical oxygen demand in seawater. In order to avoid inaccuracies, perform the following sample preparation procedure as outlined in the application sheet. First, pipette your sample into an Erlenmeyer flask. Next, add sulfuric acid to the flask, while cooling and stirring slowly. Take care that the temperature does not exceed 45 degrees Celsius. Prepare an absorber tube filled with soda lime and attach it to the flask. After 2 hours of stirring, use the M-color test, chloride test, to determine whether chloride levels are appropriate. If so, the sample may be analyzed using SpectraQuant COD tests for seawater. For detailed instructions and information, please check the pack insert. Our test kits often require an optimal pH value to ensure precise results. If the sample solution is too acidic or basic, the chemical reaction may be accelerated, slowed down, or even inhibited completely. Hence, the pH of the sample should be checked prior to starting the analytical reaction. We recommend M-ColorFast test strips for easy and fast monitoring of the pH. If necessary, adjust the pH of your sample with hydrochloric acid or sodium hydroxide, but take care that the acid or base does not introduce interfering ions or molecules. Food and beverage samples often contain colloids that must be cleared prior to photometric analysis. Carez clarification is ideal for this purpose, as it enables the precipitation of proteins, eliminates turbidity, and breaks down emulsions. Merck Millipore offers a ready-to-use Carez clarification set containing Carez solutions 1 and 2. Solution 1 is added to the sample first. Then the same amount of solution 2 is added. This causes the immediate precipitation of insoluble potassium zinc hexacyanoferrate out of the solution. It also results in the adsorption of all kinds of macromolecules, which can then be removed from the sample by simple filtration. It is important to note that Carez clarification is not suitable for all samples. Please check the pack insert for further information about fields of use and detailed procedures. We hope that you have enjoyed watching this presentation. To learn more about our test kits and sample preparation techniques, please visit our website. Here you will find application examples, further webcasts, videos, and ordering information.